Good morning, everybody, to the very first webinar from the training and capacity building activity from the very new project Health Firm, Innovative Pulse and Cereal Food Fermentation for Human Health and Sustainability, Sustainable Diets. We have three speakers for this webinar. First of all, Professor Christophe Courtin will give a very brief introduction into the Health Firm project. This will be followed by a presentation of Professor Marco Gobetti and Dr. Rossana Koda following the fermentation aspects on food composition and nutritional attributes. I will give first the floor to Professor Courtin. Christophe, please provide your introduction.
Thank you, Jan, for the introduction, and also on my behalf, welcome to this webinar. I'm Christophe Courtin, KU Leuven, and the coordinator of the Health Firm project. So I thought it was fitting to give you a very short introduction on this project before we start this first webinar. Now, Health Firm is a European-funded project, and also by CERI, the Swiss authority. We have 23 partners on board from 10 countries. We have a project that will last for 4 years. And for that, we got a budget of 13 million euros. So these are some of the numbers. And we can see all the partners that are involved in the project on the lower left-hand corner of the slide. A nice geographical representation, a lot of very good partners, or all very good partners. Now, what is Health Firm about? Health Firm is taking place in the area of the transition of animal-based products to plant-based products, and from spontaneous fermentation processes into rationally designed fermentation processes. And by looking into these aspects, we want to use fermentation to leverage sustainable plant-based fermented food production, and also make sure that the products that we can produce have optimal health benefits, and also have a look at the scientific underpinning of these health benefits, because that is something that is often missing in scientific literature today. To achieve all this in the project, we need microbial resources. We need, of course, fermentation technology. We need human health and microbiome science, and we need consumer behavior. And those are the four main work packages that we find in the Health Firm project. And if we look a little bit closer at the human health and microbiome aspect, what we try to do there, which we think is quite innovative also, is to disentangle the interaction between food fermentation microbiomes on the one hand, the food products that are produced using these fermentations, human health, but also the human gut microbiome. So these are four elements that we want to link to each other in this project, and doing that in an in-depth way, using intervention studies, using full-scale omic techniques, using plant-based materials. If you look at the layout of this project, I'm not going to go into too much detail, but you see again the four main different work packages, all the things that we are going to do. And that is, of course, backed up by project management on the one hand, and by a lot of dissemination, education, training activities. And of course, that's the reason that we have this first webinar today. And I'm very happy that Professor Gobetti and Dr. Koda were agreeable to give this first webinar and have their presentations. So with this, Jan, I would like to give the floor back to you.
Thank you very much, Christophe. If you need for the participants, if you need some more information about the project, you can find that on the website of healthfirm.eu. I do have some household details to share with you. First of all, the webinar will be recorded, so if you like to look it back and hear it back, that is possible via the website of Health Firm. If you have questions, each presentation will be followed immediately with question and answers for a few minutes. Questions can be posed in the question box. You have a question box in the webinar slides. Please make the questions selected for the organizers specifically, because if we don't have time enough and if the questions are that specific, then we can ask the presenters if it's possible to answer the questions later on. So if there are questions not answered, but they are very, very specific and you would like to get an answer, it is possible to get in contact with the presenter. Then I'd like to introduce Professor Gobetti. He's a full professor food microbiology since 2000. He is employed at the Free University of Bolzano. He is a member of the National Committee on Biotechnology, Biosafety and Life Sciences of the Italian Council of Ministers and representatives of the Italian full professors of veterinary and agriculture at the National University Council. As reported by Scopus database, he authored more than 320 scientific articles with an EH index of 64 and more than 14,000 citations. So during the last decade, most of these articles dealt with the physiology and biochemistry of lactic acid and lactic acid bacteria during food fermentation. So I think we definitely can consider you a specialist in this field, Professor Gobetti, and the floor is yours.
Thank you, Jan, for the kind presentation. Good morning, everyone. First of all, a few words. It's a pleasure, it's an honor to be part of the European project coordinated by Christoph. We are willing to participate to this project. And this morning, I tried to conjugate the fermentation mainly on nutritional attributes and on what we should do with some innovative view on the management of fermentation. First of all, we have to know that fermentation is a biotechnology process for converting mainly carbohydrates into ethanol and organic acids by mainly two groups of microbes, yeast and bacteria, in particular lactic acid bacteria. We have to know, nevertheless, that fermentation is an oxidative reduction process that our friends take for energy. Fermentation during time, although it is a very ancient process, is inspiring by the new biotechnologies, and currently we are also speaking about precision fermentation, like has been done for the medical field. Nevertheless, we have to say that fermentation is a very, very old process. There is an association in the United States that stated that fermentation probably for humans has been a great discovery than fire. Probably this is an extremization of the powerful of fermentation, but fermentation started, the historical background of fermentation started centuries and centuries ago. There is still a discussion, debate on which type of food has started before: wine, leavened bread, cheeses. What we know is that fermentation is a very, very old process that is sustainable, green, low cost, and versatile. Fermentation is a question, is a matter of numbers. There are many, many researchers dealing with the atlas of fermented foods, and a very draft estimation says that more than 3,500 fermented foods are present worldwide. And we can estimate that in Europe, our diet may account of about 20 to 50% of on fermented foods. Fermented foods and fermentation are moving from laboratories, from industries, also in the kitchen, And it's not unusual that the kitchen is the place to start fermentation, although with some risk. But consumers at the same time are aware that fermented foods may have an impact, as clearly stated at the beginning by Christoph, on the positive alteration of our microbiome and improving our. Brief introduction is just to say that when we are speaking about fermentation, we need to speak about the food system and the microbiome, the fermentation. Our microbes are the connection, the fil rouge that start, for instance, for making cheese from the farm system, from the hygiene condition of the farm and of the personnel, from technology, the house microbiota. Then this fil rouge is helpful for getting cheeses, which are obviously influence the quality of cheeses by biotic and abiotic factors. The same can be stated, for instance, if we move in vegetable products likely bread or leaven baked goods. So the microbiome, the fermentation, the microbes responsible for fermentation are already present as endophytic in plants, and all agricultural parameters and interventions may have a role, as well as technological processes, and also in this case the house microbiota, leading through the activity of the microbes to the final product, which could be a leaven baked goods, or as you want. I would like to focus part of my presentation on mainly on sourdough. 2 years ago we wrote a systematic review based on 30 years of knowledge on sourdough fermentation, and we know very well that sourdough, as an example of a natural starter, of an ancient natural starter, is extremely diverse. Just speaking about lactic acid bacteria and the former genus Lactobacillus, we may count more than 80 species, and yeast which coexist with lactic acid bacteria in the sourdough fermentation are identified as many than 80 species. Extremely diverse are also the matrices that can be used for sourdough fermentation, not only cereals, but also pseudo-cereals, legumes, byproducts, or miscellaneous products. In this systematic review, you can see on the right what they are, the main nutritional attributes which are related to the sourdough fermentation. The word cloud represents the nutritional attributes which were focused during sourdough fermentation in the last year. You can see about mineral bioavailability, glycemic index, digestibility, dietary fibers, and so on. Just speaking about some nutritional attribute which is related to the sourdough fermentation, usually leaven baked goods have a quite high glycemic index. This is a scale, a ranking of the glycemic index, which can be attributed to all. But the sourdough fermentation has the potential to move the glycemic index of leaven baked goods to high, to moderate, if not to low glycemic index, when the sourdough fermentation is combined with the addition of low amount, not excessive amount of fibers. So fermentation could be responsible for lowering the glycemic index of baked goods. We did, speaking about digestibility, we did an in vivo clinical challenge where to humans, to healthy individuals, we administered baker's yeast bread and sourdough bread with different time of fermentation. You know, For making baked goods, you can use chemical leavening, baker's yeast, or sourdough fermentation. In this case, the comparison was between baker's yeast and sourdough fermentation. So what we found was the following, that the sourdough fermentation induced higher satiety and fullness perception with respect to the baker's yeast. The orophycal transit time was faster when the intake was a slice of bread made with a sourdough fermentation, and the sourdough fermentation has the potential also to increase the content of amino acids in blood plasma, as well as the sourdough fermentation was responsible for a lower postprandial glycemia. One of the main issues dealing with fermented foods is about their role, their impact on our gut microbiome, either on the composition of the gut microbiome or on the functionality. Obviously, when you have to start such type of study, for instance, the consumption of a slice, 2 slices, 3 slices of bread per day, the use of a human challenge is very difficult because of confounding factors, because of the interference of the remaining part of the diet. In our laboratory, we had the simulator of the human intestinal microbial ecosystem, which is like an humanoid, which reproduces very well the gastrointestinal tract of humans. And you can feed with a diet, with a normal diet, also including slices of bread. So using this machine and selecting donors based on the adherence to the Mediterranean diet, we selected 2 donors which were representatives of the adherence to a Mediterranean diet. And what we did, after the stabilization of the machine, of the chyme, we started with the feeding with, also in this case, 2 comparative types of bread, sourdough bread and baker's yeast bread. And what we found that the different consumption, different intake of bread based on the fermentation had not any interference related to the microbial composition, at least at the phylum level, but almost the same information we got regarding the genus core microbiota. We found only a slight rearrangement of the main genera which are present within the machine and in the fecal microbiota. Most importantly, the different consumption, and in particular the consumption, the intake of the sourdough bread induced, as you can see in this graph, an increase of the content of free amino acids which are present mainly at the colon level. At the same time, the consumption of sourdough bread favor an increase of the content of short-chain fatty acids, which are very relevant, which have very positive effects for the human physiology. In all segments of the colon, we found an increased concentration of short-chain fatty acids, namely acetic, propionic, and butyric acid, after the consumption of the sourdough bread. Why that? Because obviously the sourdough bread, with respect to the baker's yeast bread, has a low pH, has a higher protein in vitro digestibility, has a higher content of free amino acids, higher content of peptides, and especially has a higher content of resistant starch. Resistant starch is a form of fibers that you can find in the bread, and increasing the resistant starch, you provide specific nutrients to beneficial microbial groups which are living at the gut level, which, based on fibers, they can orient their synthesis of short-chain fatty acids, so just closing So just closing the circle regarding the effect of sourdough bread on human gut microbiota. Nevertheless, I would spend a few minutes also in this still dealing with fermentation on how to manage with the sourdough fermentation. We introduce a concept when we are speaking about fermentation and microbes which are responsible for fermentation. We introduce a concept, a definition of fermentome. What is the fermentome? Fermentome is a supra entity which comprises not just the dominant starters that can be naturally present or we can add to the food matrix, but we would like to speak also about the interactions between dominant microbes, subdominant microbes, and those which are the fine satellites having a very, very low abundance. How we carried out this study? We started from sourdoughs which were stored in the first international sourdough library, which is located in St. Vita, with one of the company participating to our European project. And we started from 8 sourdoughs which were coming from different European or United States countries. We use culture-dependent approach to set up our biobank, but we also use an approach based on metagenomics, metatranscriptomics, and metabolomics. Based on the shotgun metagenomics and metatranscriptomics, we had the opportunity to distinguish between dominants, between subdominants, and satellite members, which are all present in the sourdough. Obviously, we decipher also the functional redundancy of sourdough genomes, showing that some genes are homogeneously distributed in all sourdoughs, and different sourdoughs based on differences on the microbiota composition may have a specific genes. Then the next step was the reconstruction of metabolic pathways, which are very important for this type of fermentation, which is very important for the sourdough attributes, namely starch, non-starch, polysaccharides, and sucrose pathways, pentose and pyruvate metabolism, and all nitrogen metabolism, which are important for making free amino acids. This is just an example of all the pathways that we reconstructed, but showing, I want to spend a few seconds on showing this circle. Each segment of this circle is related to one type of sourdough, and colors refer to the different microbes we found in the sourdough. So if you look just for these enzyme activities, you can find that some sourdoughs have the same genes which was present in a number of microbes. Vice versa, some sourdough has just one microbe possessing one gene. So we spoke about redundancy of genes which can be present or not in the same sourdough. Based on this first part of our research, we set up a synthetic microbial community. The synthetic microbial community was comprising dominant microbes, dominant species, subdominant species, satellite species, and dominant of lactic acid bacteria and other bacteria, and dominant yeast and subdominant yeast. This synthetic community was compared to a natural sourdough where we know the microbial composition. So some information, when we look at the transcriptional level, you can see that dominant microbes which are present in the sourdough have the expression of genes which didn't change with respect to the presence of other microbial partners. Some subdominants have variation depending on the partners, and satellites show a great variation depending on the partners. But what we found was that dominant are important, but subdominant members of the sourdough process have unique gene and transcript and enzyme activities which were not present in dominant microbes. This was true for subdominants, but this was also confirmed in satellite members. The conclusion was the following. In our opinion, it is important we control, we steer the fermentation looking to dominant, but at the same time, we need to pay attention about the metabolic networking and about the presence of subdominant and satellite members. There was a confirmation. In the synthetic community we reconstructed, the number of I want to say the transcriptome redundancies was much higher than in the natural sourdough. But especially if you were looking, we used the synthetic community 40 days with refreshments every day using durum flour. And this was compared with the natural sourdough. What we found was the following, that the synthetic community kept all microbes except of one yeast for 40 days, while the natural sourdough we have used change almost radically the microbial composition. So stating that we need to find the right compromise, the right association, the right metabolic networking among members. One explanation was the following. We were looking to the volatilome profile of the synthetic microbial community, this one, and of the spontaneous sourdough. This is the volatile profile at time 0, and this is the volatile profile after 30 days of refreshment, which means, based on this statistical elaboration, this principal component analysis, that the volatilome profiles were markedly different. This is the situation regarding indeed the synthetic microbial community, which after 30 days of fermentation was almost the same, showing once more the importance of the metabolic networking. Coming to my conclusion, I want to leave you with this slide we use in a review we wrote several years ago. Fermentation is obviously an ancient process which know very well since centuries, but based on our knowledge, based on the availability of many techniques we have already available, as I showed you, homic techniques, nevertheless, fermentation is likely a metabolic labyrinth where we must be very skilled to carry out the most suitable combination of microbes and the most suitable parameters to get the most suitable nutritional attributes and combination among microbes. Thank you all for the attention.
Thank you very much, Professor Gobetti, for this really interesting but also very comprehensive and short overview on fermentation. I am actually a little bit surprised that I don't see any questions, but your presentation does raise some questions for me, because there are quite some things I would say that is interesting to know, especially if you look at the purpose of the project as such, because Health Firm project is intending at the end to produce products on a precision fermentation technology basis, and at the end we would like to be able I'm just looking from a company perspective to be able to say something about such products. Taking your presentation into account with the variability of, at the one hand, the microorganisms that we can use, and with the diversity, how they can be combined in relation to, amongst others, also the diversity of the sources that we use to be fermented, like wheat and whatever you can use. How far do you think that it's possible to make claims on these kind of products, like what the industry would like to do, health claims, having in mind the requirements that EFSA is using to review the dossiers that food industry is submitting?
Thanks for the question, Jan. With respect to 20, 30 years ago, claims are more restrictive, need scientific proofs, and as you have seen, I think that fermentation is a powerful technique to show benefits for human. As we are doing in this European project, obviously you need some confirmation of what you see in your laboratory, what you see using the machine, as I showed you, the simulator of the gastrointestinal tract, with a number of human challenges. If you have a look on the literature, also starting from fermentation, you can see an increase of the publication dealing with the human challenges to have the final proof, which must be the key point for industry to claim, for instance, a low glycemic index, an increase of the digestibility, an increased content of bioavailable, bioaccessible minerals. So we are on the route. We have skills, we accumulate data. We obviously need time to develop human challenges which can allow the claim to industries.
Thank you for the answer. I now see some questions being posed by the people, and one of those questions is, for instance, there is a large difference between bakers using sourdough products that are sold in the bakeries and using specific strains and fermentation conditions. So it's about strains and conditions as well where they are fermented. How are we going to match these two in the project of HealthFirm?
I think the last part of my presentation was dealing with an approach where we probably have to change a little bit our way for steering fermentation. Steering the fermentation for decades was based just on checking on dominant starters. On those starters that we are using, we are adding to the raw matrix, to the food matrix. Now we have to pay attention on all the consortium that is present. Obviously, if we compare baker's yeast fermentation with respect to the sourdough fermentation, I think we have with sourdough fermentation a lot of advantages with respect to the baker's yeast. The only drawback probably is the more complex fermentation ecosystem and the time of fermentation, with in some cases in an industrial process cannot be so much tolerated.
Okay, and another question that was posed is about the fungi that are usually also present in the sourdough communities. You focused first of all on the lactic acid bacteria in the sourdoughs. What is the contribution of the yeast that are present in the?
You're right. You know, the performance, the power of the sourdough is because of the presence of lactic acid bacteria and yeast. Although we have to say that in some country you have a sourdough with only lactic acid bacteria. In my opinion, although I dedicated most of my research activity on lactic acid bacteria, yeast are indispensable for the synthesis of carbon dioxide, for the leavening of the dough. They have a contribution, therefore, on the rheology of the sourdough. They give a contribution also on the synthesis of some volatile compound which is responsible for the flavor. Yeast are also responsible. In some cases, there are yeast who have very important enzymes also for nutritional attributes, and yeast in some cases are also responsible, together with lactic acid bacteria, for the extension of the shelf life of the sourdough leaven baked goods. So they are in synthesis, they are also important, right?
Yeah. And at this stage of the project, do you already see any gaps in the microbiome research, fermentation, biotechnology, biobanks, legislation that is a struggle for you even at this stage? Is it, and if that's yes, would you see some ways to tackle this?
Yes, probably I'm not the best representative to say something about that, but I want to mention that I was so lucky, I was honored to be one of co-author of a scoping review, which has been recently written by the group of the coordinator of the European project, Professor Christoph Karting. And this review approached this issue. So we have to define at the legislation level what is the sourdough, what is the starter that we can use for making sourdough, in view to guarantee the consumers that they are buying a bread, a baked good, which has been made really with sourdough. And what I can state is that there is a lack of legislation, and there is not an homogeneous legislation among countries in Europe. There are differences, for instance, between Belgium, France, Spain. For instance, in Italy, there are at least 10 years that we are trying to define a legislation for bread making, but we were unsuccessful and we have a lack of legislation. You touch an important point, and I want to mention once more the contribution of this scoping review from the Christoph Karting group.
To just a question that pops up with me is, to what detail do you think that legislation then should describe the process and the outcome of what, for instance, bakers with sourdough are producing?
When we are speaking about the legislation, I think we must be careful. We must be able to find compromise between what the purpose of companies, the guarantees of consumers. I don't think we can go so much in detail, but at least to define what is certainly made with the sourdough and what is not.
Yeah. Okay.
You cannot in synthesis, you cannot go so much in detail.
Yeah. Maybe a final question, because I know that your schedule is rather tight. Is in your opinion, do you think that the substrate, so the sources that you use of fermentation, at the end determine the metabolites, or is it the composition of the microbiota that determine the metabolites?
Both. And you know, the same metabolism may approach different substrates in a different way. If you are using, you know, food matrices are providing different substrates, and obviously in the presence of different substrates, the adaptation, the reaction, the metabolism of the same microbes may change, especially on those microbes which have a lifestyle as free living or nomadic, meaning that they can stay in different food ecosystem. You can exclude neither a substrate, neither microbiome composition. The powerful fermentation is from the most suitable combination of substrate and microbiome.
Thank you very much. A very, very last question to you. You referred in your previous question to a study that was in a video, and it's questioned if you can share the link to that study in the chat box so that people can attend that link to see what is described there.
For sure, it will be a pleasure. For sure.
Thank you very, very much, Professor Gobetti, for the excellent talk.
Thank you.
And I'm now going over to Dr. Rosanna Koda. She is currently a PI and adjunct professor at the University of Helsinki at the Department of Food and Nutrition. She has a background in food microbiology, focusing on biotechnological applications of lactic acid bacteria again. Like Professor Gobetti, she has been working with fermentation for more than 15 years, so she has a very, very nice background, and in particular on nutritional and technological improvement of plant-based foods, and research interests encompass study of food microbiota and the use of microbial metabolism to create innovative solutions for the agro-food chain and to contribute to a sustainable transformation. And that, of course, is a very excellent ending of your short bio. Dr. Rosanna Koda, please, the floor is yours.
Thank you, Jan. I hope you can hear me well, and good morning to all the attendees. So this will be part 2 of this webinar, in which I'm going to discuss about the impact of fermentation on the chemical composition and some potential nutritional attributes, but also on flavor properties of fermented food. So we have heard from the previous presentation how much fermented foods are strongly connected with people's tradition all over the world. They are important for us, they are part of our cultural background. But we need also, nowadays, to bridge the past with the future. And specifically, we are assisting to an expansion on the market of fermented food that are based on plant and that are aiming at substituting their animal-derived type of food. So this is an important issue that we are trying to focus on. On top of this, we need to consider that fermentation still remains a technology that can be used to completely create and innovate food products. Last but not least, we've also heard this before, there is an increasing evidence of the positive effect of fermented food on gut microbiome, and more studies are certainly coming rather soon. Today, I will focus my presentation especially on the challenges of fermenting plant-based ingredients. And here there are a few of these challenges. We know that plant-based ingredients, legumes and other type of grains or side streams, they might have some challenges. They can have some so-called anti-nutritional factors. A few of these, like phytic acids, different type of phenolic compounds like tannins or trypsin inhibitors, might hinder the digestibility of proteins, for example. Other types of anti-nutrients are raffinose oligosaccharides. They are very common in many types of grains, and other are specific of specific legumes like vicin and convicin in faba bean. We also have some issues in terms of technological performance. For example, flavor issue. Some grains, some legumes, they can have this beany flavor. They can be bitter, and they might have also a poor structure forming capacity, especially compared to, for example, animal proteins. All of this brings to low consumer acceptance, which might be a problem if we are trying to increase our consumption of plant-based food. Fermentation is a technology that can be used to add value to plant-based food. How? We see this in the enhancement of nutritional properties. We heard before that, for example, we can decrease anti-nutritional factor and as a consequence increase, for example, starch and protein digestibilities. We can aim to the synthesis of bioactive compound. Fermentation can also be used to improve technological feature. We can modify the virological properties, for example, via the synthesis of hexapolysaccharide, but we can also target specific flavor design. Last but not least, fermentation can control undesired microbes and standardize the quality. It is the reason why it has been used for centuries, and it is still an important feature. All of this can bring to more wholesome food with attractive properties. From tradition to innovation, why is that? Well, here you can see a figure representing the history of production of Parmesan. This is a food that is known all over the world, and we know a lot about it, of history and microbiology and many other things. What is the challenge now is that we are trying to produce similar type of food that is plant-based and that we hope that it possesses the same flavor and nutritional attributes. And I like also to show how fermentation can be used in a completely new and creative way, like in the examples that you see here, this mushroom garum or this wild rose vinegar. They are products produced by the famous Noma in Copenhagen, and they are based on fermentation. So how do we get to that? Well, microbes have a paramount role in shaping the properties of fermented food. And when we think about food fermentation starters, we know that they can be selected based on their metabolic traits. And the metabolic traits rely on the genomic information of the strain, but also on the environmental pressure, for example, the specific food ecosystem or process condition that we want to use. So the interaction between starter and raw material is very important, and it is what defines the quality of the fermentation. Nowadays, we can also consider that to increase our success and to drive our results, we can also use many technological aids, for example, fractionation of the raw material, germination, and even the use of enzymes. They can help facilitate fermentation. I'd like to show you a few examples in this part and to discuss about potential nutritional properties that we can achieve via fermentation of plant-based food. I want to discuss a little bit about fermentation of faba bean. It is a type of raw material with which we have worked quite extensively in the past few years, not only in my team, but also at the European level, because it is a crop with recognized importance and it is considered as domestic source of protein. So in a study a few years ago, we have done an experiment. We have used faba bean flour and we performed fermentation, that's so-called sourdough type 1. And that is the type of artisan fermentation of sourdough that would be done in, for example, a small artisan bakery. So we have propagated faba bean flour in this way, and we carried out a number of backslopping steps until the sourdough would be mature. And we have seen that this backslopping protocol enhanced the nutritional attributes of faba bean. For example, it increased total phenol's and antioxidant activity and decreased anti-nutritional factors. Throughout the backslopping, we also studied the evolution of the microbiota in the faba bean sourdough, and we saw that it quite resembled the microbiota of kimchi. We found many Pediococcus, Leuconostox, and Weissella. And all these lactic acid bacteria strains were further phenotyped, and we wanted to see indeed what potentially beneficial technological feature they could have. Like, for example, alpha-galactosidase, beta-glucosidase, peptidase, phytase, and so on. And a few of these starters have been used in further study as starters for faba bean and other grains fermentation. Talking about the degradation of anti-nutritional factors in faba bean, in one study we tried to see if by fermenting with lactic acid bacteria possessing beta-glucosidase activity, we would be able to degrade these compounds, vicin and convicin, who are the precursors of the toxic divisine and isoramyl that are responsible for a disease called favism. Favism is a form of genetic disorder that affects some people, and it's a sort of hemolytic anemia. And via fermentation, we were able to almost completely decrease these toxic precursors, and the toxic compounds were not found anymore. And the ex vivo confirmation of toxicity was also carried out. In another study, we also wanted to see if in more mild acidification condition, we would achieve the same result. And we particularly applied this fermentation to production of bread. So we noticed that there was certainly a decrease in vicin and convicin. They were stable in bread, and luckily the toxic aglycones were not detected anymore. However, fermentation conditions are really important to achieve the desired result in this respect. The same faba bean sourdough bread was also evaluated for many other properties, and here you can see a picture representing the results that we obtained. So generally, the addition of legume in bread to a certain amount might confer some undesired effect. However, the use of sourdough fermentation was able to at least partly restore the technological feature. But what was especially important was that we noticed that faba bean sourdough had better nutritional properties, conferred better nutritional properties to the composite wheat bread. We observed an increase in the protein digestibility, a decrease in the glycemic index, and generally the nutritional quality was overall better. After all, the porosity of this bread was quite okay compared to the control, and the sensory properties were acceptable with at least the absence of flavor. In a similar approach, we also performed fermentation with this Lactobacillus plantarum possessing beta-glucosidase activity, and we used this time this fermented faba bean as ingredient in pasta production. We noticed similar trend of improved nutritional profile with higher protein digestibility, higher protein biological value, and improved amino acid profile. So this was an important result because, for example, this can represent a way to use a fermented ingredient to produce more innovative food. Pasta is certainly not our typical fermented food. And this is also one important strategy to fortify cereal-based food with legumes and to complement the essential amino acids of the two crops. What's more, we can also target fermentation to obtain even further functional benefit. There are two examples here. In this study, we use an association of two lactobacilli with specific peptidase activity, and we fermented very different legume flours. And we obtained the formation of lunacin-like polypeptides. Lunacin is an anti-cancer peptide with also antioxidant properties. And we were able to achieve this formation in different legumes, and especially in beans, chickpea, and pea. And in a recent study, we also used an association of Propionibacterium fernandriki with Lactobacillus brevis to increase the vitamin B12 content of this fermented legumes flour. The result was very positive. Vitamin B12 was produced in sufficient amount to be useful potentially in the daily diet. And this is important because plant-based raw material is lacking in vitamin B12. Coming to another substrate, this is an example of side stream production from the cereal industry, which produce very abundant amount of different type of side streams, from wheat bran to also brewer's spent grain. Fermentation technology can be used to functionalize side stream, and this could be a very important result, because typically this side stream possess a few technological challenges. They have a very high fiber content. It might be difficult to implement them at sufficient amount in different food applications. Sometimes they have not very pleasant taste, and they generally have poor structure forming capacity. Functionalizing them via fermentation is a good way to achieve improved nutritional quality, which generally is quite good in this side stream, but also to implement a more sustainable food chain. There are a couple of examples of this approach here. In a study a few years ago, we wanted to see if it was able to improve the bioaccessibility of proteins from wheat bran. Wheat bran is quite a good source of protein. However, they generally remain entrapped in the complex structure of wheat bran. And we used, in this case, fermentation with consortia of lactic acid bacteria and yeast, plus some cell wall degrading enzymes, especially xylanases. And we were able to solubilize 50% of the entrapped proteins, and we saw an increase of the in vitro protein digestibility. Another interesting feature were also improved, like increase of phytase activity. In a follow-up study, this bioprocessed bran isolate was used to as bread ingredient, and we noticed that it was able to confer softer bread and to delay the stalling compared to non-processed bran protein isolate. With a similar approach, in a more recent study, we also bioprocessed brewer's spent grain. In this case, with a combination of xylanase and a fermentation with Lactobacillus plantarum, selected to increase the antioxidant activity of brewer's spent grain. So we obtain a release of phenolic compounds, but also bioactive peptides were formed from encrypted sequences in the barley and corn protein of this spent grain. And lactic acid bacteria were also able to further convert phenolic acids into more active form. When this fermented spent grain was used for pasta, we had a technological positive effect because we noticed a more homogeneous protein network, but the antioxidant effect was still available after in vitro digestion of pasta, as this was confirmed in some ex vivo cell assay. And concluding the part on the fermented side streams, there is more that can also be attempted. For example, oilseed pressed cake can also be fermented, and rapeseed protein concentrate is the case reported here. We have used fermentation with a dextran producing lactic acid bacteria, and the rapeseed protein concentrate used as ingredient in bread was able to restore the quality when no fermentation was done. And also we observed an improved free amino acid profile and decreased raffinose oligosaccharides as consequence of the fermentation. And in a very recent series of studies, we also have used fermentation to improve the properties of a bread slurry. This bread slurry was deriving from surplus bread that is generally waste in bakery industry. It's a sort of side stream. Generally, bread slurry, if used as such in bread, is mostly detrimental when it is fermented with the right conditions and right starters. For example, we can achieve synthesis of dextran, which is able to restore then the quality of the bread. And in other two examples, we have also fermented with a specific lactic acid bacteria strain. We're able to achieve antifungal peptides, and this ingredient performed as a natural preservative in bread application. Or even in another case, we were able to improve potential nutritional properties via, for example, the biosynthesis of GABA. So part two would now be effects on technological quality and a few examples on texture and flavor improvement via fermentation. So why it is so important to consider flavor and structure of plant-based raw material? Well, flavor active compounds are very abundant in plant materials and might be challenging when we are trying to develop dairy alternatives or meat alternatives. And clearly, flavor is an important limiting factor of the acceptability and marketability of this new plant-based alternative. So what are possible solutions if thinking about fermentation technology? We did a recent literature research, and we have observed that most typically the most common approach is fermentation with lactic acid bacteria alone or in consortium association with yeast. And another possible way to improve the flavor of plant-based raw material is also the synthesis in situ of exopolysaccharides. Exopolysaccharides are very important extracellular polymeric substances produced by microbes during fermentation. What you see here are a very famous series of examples. This is what we can find in yogurt. These are kefir grains or even the SCOBY of kombucha. Exopolysaccharides also have important technological advantage that we can use. For example, they are natural texture enhancers. They can, when used in bread, confer a higher specific volume and confer more softness. They can improve sensory properties by improving the mouthfeel. They can mask of taste, and they can also have some potential probiotic effect. One of the exopolysaccharides that has been most studied in this past year, especially in bread making application, is dextran. This is due to the many benefits that it can give. It is a natural hydrocolloid with water binding capacity that can modify water distribution and stabilize the solid liquid interface. Dextran are synthesized by a dextran sucrase enzyme produced by, for example, several species of lactic acid bacteria. And when specific sugar acceptors are present, like maltose, for example, also oligosaccharides can be formed. So there is quite a biodiversity of dextrans, and different strains have different ability to synthesize dextran, and dextran properties also depend on their molecular structure and interaction with the environment. And it has been observed that linear dextran of high molecular weight is the best performer in baking application. And I want to show you a few examples of this. Here you can see some sorghum bread, and the presence of dextran has improved the technological quality of this sorghum bread. In that specific study, we also noticed that the presence of dextran was indeed able to improve very much the mouthfeel of dextran sorghum containing bread, and also bitterness was very low and almost not perceived. And this would be an issue in producing a bread of this type with a high percentage of sorghum. A very similar result was obtained again with faba bean, when the presence of dextran significantly improved the quality of the bread and also the flavor profile. And these breads indeed look very different from the other breads that I showed before. Another example that was very recent is also the use of a similar approach in baking brewer spent grain with composite bread. So the presence of dextran improved significantly the quality. And in that specific study, we also studied the effect of dextran containing brewer spent grain bread on the in vitro gut metabolites. So we noticed that there was an improvement in free amino acid bioaccessibility and lower ammonia was produced. This is important because we can implement in the bread recipe more of this nutritionally very valuable but challenging raw material. And we can increase overall the nutritional properties of bread, more fiber, more protein without negative repercussion. And going to a completely different food model, this is the case in which the same approach was used to produce some meat analogs. So our intention was to use dextran production in faba bean protein concentrate, that is a rather challenging raw material, is particularly bitter. And we wanted to process it in a way that we would obtain dextran enough to mask bitterness, but to avoid excessive acidity. So we have used, in this case, modeling of the fermentation parameters. We have introduced a few factors of interest like time, temperature, flour water ratio, inoculum, and we measured the responses, dextran, viscosity, and acidity. And we were able to find some optimal fermentation condition to be used to process this faba bean protein concentrate. In a follow-up study, we have then evaluated the effect of this tailored, optimized fermentation on the production of these extrudates. What you see here are faba bean protein concentrate and gluten extrudate, and we have done carried out a sensory study to evaluate that, in which different samples were compared. There was the optimized fermentation, there was fermentation without dextran, there was a control with no treatment, and there was also enzymatic dextran used to produce this meat analog. So the descriptive sensory analysis was able to confirm that dextran in optimized fermentation, but also added as enzymatically produced, was able to effectively improve the sensory properties of the extrudate, and we have observed decreased bitter and pea flavor, sourness was low as we wanted to, but also an increase in sweet and umami taste. And in the last food model of today, we'd like to also consider how, for example, exopolysaccharides technology in fermented plant-based drink can be beneficial, or this type of yogurt product. Fermentation can also, in this case, modify flavor attributes, and the synthesis of exopolysaccharide can be considered beneficial because it can help to produce stronger gels and to give more efficient water binding. The current approach is to effectively use dairy yogurt cultures and probiotics, and this is quite a successful approach so far. However, we can also consider that we can, if we want, tailor more specific properties during fermentation. In a study of a few years ago, focusing on emmer beverages, we noticed that different starters were able to confer indeed different type of properties to this fermented drink. And this is also the case observed in literature, where fermentation process can be adapted to each plant raw material in different way. And different studies are available on lupine, quinoa, grain and legume mixture of different type. This what you see here is recent studies. An example of our Lactobacillus brevis, for example, was able to produce exopolysaccharides during the fermentation of oat bran and hemp protein concentrate. So to conclude, fermentation, back to the future. We are now in a moment in which we want to harness what we learned in century of fermentation, and we want to apply our learning to create and to solve some of the challenge of our time. Fermentation provides many opportunities to achieve more appealing plant-based alternative and to create new plant-based food. This would contribute to more sustainable diet. Our tools to do this are an increasing integrated knowledge of the fermentation microbiome. We've seen this in the previous presentation. And this is one way in which we can achieve a rational design of fermentation consortia for multiple nutritional benefits. And I'll conclude with a sentence again from the fermentation head in Noma Copenhagen, that they have inspired my presentation. And in this sentence, which I share, fermentation is not just a trend, it's here to stay. And the increasing understanding of the complexity of fermentation can really be a valuable tool to shape the food of the future. And last but definitely not least, I just want to give my deepest gratitude to all my colleagues at the Department of Food and Nutrition at the University of Helsinki and the Grain Technology team, to all the many partners in these years that have contributed to the projects that I have discussed about today, VTT and University of Bari, University of Bosen and Sapienza, and of course to the funding organizations that have supported our study. And last but not least, I would like to really thank you all for listening. Thank you.
Thank you very much, Dr. Koda, for the excellent presentation. A very nice overview. And I understand that there are some people that have raised some questions. So I have to pick them here to read them. First of all, there was a question about the fact that the effects of experimental sourdoughs used in laboratories, they differ from the sourdough used by bakeries. That was summarized in a very recent systematic review in Advances in Nutrition. As a result, health benefits claims for common spontaneous sourdoughs have not been substantiated. Actually, will the experimental ferments used in the projects be adopted by the market? What do you think about the, let's call it the commercialization options of the output of the project?
In reference to health firm, do you mean? In
reference to health firm, yes, obviously.
I think that indeed this is one of the points that we want to address in the project health firm, because the idea is that when we are able to study and understand how specific consortia would perform in different fermentation, one of the possible outcomes is that these starter cultures could be made available for further industrial use. So this is something that we are also trying to work towards.
Mm-hmm. Maybe I'm now a little bit late to Professor Courtin. Maybe I can invite Professor Courtin as well to switch on his camera and his microphone, because these are obviously also questions that we could address to you, Christophe. Apologies for being late to invite you to switch on the camera. What is your opinion on this specific question? Commercialization of the end products of results of a health firm.
Well, I think in first instance, if you look at the intervention trials that will happen within the context of health firm, these will be also on industrially produced products. So we will have laboratory scale fermentations, we have pilot scale, and we have industrial scale fermentation. So for sure, we'll take that in account. And as Rosanna also mentioned, yeah, we have that overlap between the different levels. So I think we'll learn a lot from that. And that there's differences between scientific practices and industrial practices. Well, I think that's clear also.
Yeah, of course. Thank you. Another question was about gluten-free products. That's very specific, of course, but there's let's call it a trend to grab gluten-free products. It has not been passed by, but what is the consequences of fermentation technologies and gluten-free products? Rosanna.
I think I can say quite safely that, for example, this dextran technology has been widely used, at least in literature to show how fermentation can contribute to create, for example, baked goods with more appreciable quality in that respect. So fermentation is the technique that can be tailored in many different ways to achieve a series of important results, also for gluten-free type of food.
Mm-hmm. And in that respect, there's one of the participants that would love to hear the perspective on substituting faba bean and the faba bean fermented outputs and its potential effect on organoleptic properties of health-oriented industrial products. So how implementable is fermented pulses and legumes in cereal-based products?
I think in my opinion, and I think I share this opinion with the vast majority of my colleagues, this is definitely one of the goals we are working for, but just saying the word fermentation is not enough. We need to think about what type of fermentation we are doing for what purpose. We can tailor flavor profile with fermentation in different ways. So it is certainly a way to follow also for this.
Yeah. So application of pulses and legumes is definitely a very good replacement of cereal sources. There are quite some questions about very specific things like production of B12 by propionic bacteria. What is the effectivity of so how much of these nutrients that you usually can't get from plant-based foods that are limited in essential nutrients? How can fermentation technologies contribute to the intake of these essential nutrients?
Definitely, this is another one of the aspects that concern especially improvement of health properties of plant-based fermented food, towards which we are working in this health firm project. And it is biofortification is a potential way to achieve also this result. In the specific study that I mentioned, we were able to produce a significant amount of vitamin B12. And there is, if I remember correctly, also some calculation that if used in bread at this much amount of sourdough, and if I eat one slice of bread, how much of vitamin I could get. So some theoretical assumption has been done. What would be important also to see is what happens to the fate of this vitamin produced by fermentation when implemented in food recipe, and how they could be bioaccessible. So I hope that we would be able to see also more of these results, because fermentation-wise, this is a possibility indeed.
Okay, so fermentation is a very nice tool actually to increase these limited nutrients. Christophe, another question that was probably better be answered by you as a project leader. Is do you have any idea of the fermented products you'll target at in the project? So how many, yeah, and what types? How many? What types?
Not the amount. So we will be looking at solid products like sourdough bread, for example, tempeh, texturized vegetable protein type products. That's one. We will look at liquid products. So let's compare it to oat drinks and others. And you could say, well, oat drinks are on the market for sure. But what we often notice is that protein levels are quite deficient. If we look at fiber content, it's quite low, while oats, for example, have an enormous potential to provide these. So one of the aims of the Health Firm project is also to develop the technology to make sure that we have optimal use of the nutrients that are available in, be it oats, be it wheat, be it faba bean or yellow pea, which are the four main substrates that we will be looking at. So drinks also, and then semi-liquids or semi-solids, whatever you want to call it. It's like yogurt type materials, where again, we can use fermentation technology to have that solidifying or gelling aspect. The exopolysaccharides were mentioned, but also again, looking at protein functionality, you can do a lot with fermentation to functionalize protein, starches, fibers, and so on. So we will be looking over all these different categories of food products.
Thank you. Another interesting question is actually, is it viable to consider fermentation technologies not only in the direction of health benefits, but also to reduce as a strategy to reduce food waste? Maybe Rosanna can.
Yes, I like this question, and I very much believe that this is the case, and this is has been the aim of a few recent projects we have had. Not only us, but this is also reported in literature quite widely. Fermentation of brewers spent grain and how to reintegrate it in the food chain. I presented the case even of surplus bread. There are other studies on that, and other byproducts are certainly available even in literature. So to have this sort of upcycle food, the technology is very promising. We are able, and we will be able to do it better and better. We need just to understand how at the food system level we can implement this efficiently for every actor of the food chain, because side streams are side streams. They are not the mainstream after all.
Yeah.
If I may add on to that, so I think shelf life is also a very important aspect. So if we have fermentation technology, we can enhance shelf life. That means that the possibility that a product is eaten before it's actually has to go into the waste bin is much larger. So I think we have to make people understand that shelf life is a large aspect of sustainability also, because longer shelf life means less waste, means more sustainable use of the foods.
Yeah.
And so I think side streams, yes, and also the shelf life aspect is very important, and their fermentation, of course, is quite important.
In that sense, another question is of scaling up the fermentation technologies in such a way that it's standardized, controllable, and that kind of thing. So that's how will that be tackled in the project?
Well, I think fermentation technology, if you look at the dairy industry, for example, it's being performed at a huge scale. Huh? So part of that technology will be transferable to plant-based materials. Some of it will be not transferable, but I think we have a lot of very clever minds in the project, both from academia, institutes, as well as the industrial field, that we will be able to tackle that transformation from dairy-like products into, or dairy-based products into plant-based products, or meat-based products into again, plant-based products.
Yeah, you refer to the dairy industry, and the dairy industry I know from the past, because I worked there, that the sources, the milk, are very much controlled on contaminants. For the plant-based foods, that's of course another issue. So there's also a question about, can fermentation technology reduce contamination of potential sources that are used for fermentation?
My answer would be yes. This is one of the aspects that would that should also be used when thinking about fermentation. It can be a way of standardizing the quality. It is not the easiest way, but it is certainly a way. And it is one, after all, fermentation has been defined as the oldest preservation technology of food. So this is something that can be also envisioned and implemented in this respect.
Even if it's about pesticides and that kind of things?
We are talking about agricultural practices, and yes, but I do not have a single answer here to say, oh yes, we can reduce pesticide or we can obtain specific outcome with fermentation. But the point is that, in my view, the food chain should cooperate broadly at different aspects if we want really to achieve the results of a more sustainable food system. It is difficult to give just to fermentation technology every possible input that we need to implement a good and healthy, balanced diet and sustainable food system.
Yeah,
I would second that, because it's fermentation is a promising technology, but it's not a magic bullet to solve all the problems in the world. So I think pesticides, mycotoxins, that sort of thing, we should try to control it in first instance at the primary production level. So let's see that, and the fermentation can help maybe a little bit, and even then, but we should not rely on that for these aspects of mycotoxins and so on, or pesticides.
Very practical question. Is the project recruiting postdocs?
I'm sure, I know that there are partners that are still looking for people, whether that's PhD or postdoc, I'm not entirely sure. But what I would like to suggest is that people that are interested in the project and also in vacancies that might be related to the project, that they would keep an eye on the LinkedIn page of HealthFirm. Because if we have job openings on the project, they will be posted there. And so interested people can always find information back there, next to, of course, results from the project, partner information, and so on and so on.
Okay, having said that, Christophe, Rosanna, and Marco Gobetti, who has already left, unfortunately, because due to other obligations, I very much would like to thank you for giving these presentations, and thanking also the participants, because we had quite a large audience, to be very honest. It was rather successful, I think. I will make an announcement as well, that please take a look again at the LinkedIn page of HealthFirm, but also at the HealthFirm website, because we aim at several webinars this year, workshops this year, that mostly will be free attendable. So you're invited to participate there as well. Thank you again. And with this, I'd like to close this very first webinar of the HealthFirm project. Have a nice weekend.
Thank you very much. Thank you.
Bye, everybody.